Hi, I'm Brenda, and I'm going to talk about the atomic force microscopy, AFM. The atomic force microscopy is a high-resolution, not optical, imaging technique first demonstrated by Binet, Quaid, and Herbert in 1985. Since then, it has developed into a powerful measurement tool for surface analysis. AFM allows accurate and not destructive measurements of the topographical, electrical, magnetic, chemical, optical, mechanical properties of the sample surface with very high resolution in air, liquids, or ultra-high vacuum. The instrument uses a cantilever with a sharp tip at the end to scan over the sample surface. The AFM tip is usually made of silicon or silicon nitride and is integrated near the free end of a flexible AFM cantilever. A piezoelectric ceramical scanner controls the lateral and the vertical position of the AFM cantilever changes. As the probe scans over the sample surface, attractive or repulsive force between the tip and sample, usually in the form of van der Waals forces, but also can be a number of others such as electrostatic and hydrophobic interphilic, cause a deflection of the cantilever. The deflection is measured by a laser, which is reflect of the cantilever into photodiodes. As one of the photodiodes collects more light, it creates an output signal that is processed and provides information about the vertical bandit of the cantilever. This data is then sent to a scanner that controls the height of the probe as it moves across the surface. The variance in height applied by the scanner can then be used to produce a three-dimensional topographical representation. What can I say with AFM? In that case, I have a different kind of images and I explain some of them. I explain the letter V, the letter E, the letter H. In the letter V, you can see the cyclic nucleotide regulate potassium channels reconstituted into lipid membranes. In the letter E, you can see an imaging for a living cell's A2 cell building and pulling collagen fibrils cutting a substrate. To maximize the construct, the exemplified imaging shows the deflection of the cantilever, which changes while contouring the sample. In the letter H, you can see the circular plasmid DNA imaging in buffer solution by frequency modulation AFM. Red and blue arrows indicate major and minor grabs of the DNA respectively.
Hi, this is Wendy and I'm going to talk to you about cryo-electron microscopy. First of all, we need to know that this is an imaging technique that was developed by Jacques Dubochet, Joachim Frank, and Richard Henderson. And thanks to this invention, they won the Nobel Prize in 2017. Let's talk about the cryo operation principle. It generally consists in taking a sample, then freeze it with a special liquid such as ethane. Once the sample is frozen, you load it into the electron microscope. An electron beam processes the sample, and now the molecules do not get damaged thanks to the freezing step mentioned before. Once the electron beam hits the molecules, they leave a unique shadow, as we can see here. These unique shadows are detected by a camera. A computational algorithm detects the shadows that have the same orientation, then puts all these images together into one. This process repeats for every molecule orientation. When the program has all the images orientation, it compacts them into one. Finally, by reconstruction, the computer gives you a three-dimensional image. Here we can see some examples of the images that Cryo-EM provides. We can see here the Zika virus structure And we can see here the gamma secretase structure. Now you might be wondering, yeah, we have a three-dimensional image, and then what? Well, thanks to the three-dimensional image provided by Cryo-EM, researchers can see the form and structural features of the molecules and then discover the way that the molecule works. This is helpful in pain treatment and cure diseases. For example, I found that the surface of SARS-CoV-2, the virus that causes COVID-19, was revealed using cryo-electron microscopy, and due to this, researchers could learn the mechanism of infection and then study possibilities for vaccine and treatments. Now we're going to discuss the main differences between cryo-EM and AFM microscopy. One of the most remarkable differences between cryo-EM and AFM microscopy is the physical principle that each one uses. The Cryo-EM works with an optical principle in comparison with AFM that is a non-optical technique. Consequently, the operation principle of each one changes. While the Cryo-EM uses an electron beam to hit the particles and then analyze the shadows they leave, the AFM image is formed thanks to the repulsion between the sample and the cantilever. Also, we have to mention that in cryo-EM technique, the sample has to be covered with a special liquid so that the sample freezes. Finally, something we must be aware of is the information each technique provides. The AFM gives us a topographic map, the electrical, magnetic, and chemical properties of the sample. By the other hand, the cryo-EM provides us with a three-dimensional image of the molecules, and due to that, researchers can learn how the molecule works.
To talk to you about the bionic applications. So the IFM allows to study the microstructure of materials, allowing to study their physical properties such as viscoelasticity, electrical force, magnetic force, hardness, homogeneity, etc. This allows the AFM to study mainly four types of samples: polymers, ceramics, metals, and biological. We know that metals, polymers, ceramics, and composites were and are fundamental pieces in the development of science and technology. But from 1990, work began on another alternative, such as biological materials, whose properties generally exceed those of the traditional materials and have a greater ability to be multifunctional. However, not all the applications in which they could be applied are known. This is because science is still in the science and production of these materials. That is why bionics has a great impact in this area, either for better study of the structure and properties of these materials, for their application or study natural biological structures and synthesize new materials from these. Some of the most significant examples of these materials are spiderweb, red abalone shell, and blue mussel glue. The spiderweb, for example, is a natural biological material that has a very high potential and can be used in different applications for your properties. An example of this potential is that many material synthetic fibers, such as Kevlar and polyethylene fibers, very high density, rich moduli and tensions very high. However, being composed of the crystallinity very high, these fibers tend to be brightly and not very strong when under pressure. However, spiderweb, despite not having modulus of elastic, has some synthetic materials, has a high elongation at break, and is stronger under compression. Therefore, in this type of materials could be applied in the artificial would be a very useful scientific advance, and for this one of the best tools that bionics can use the atomic force microscopy. In nature, there are not only natural biological materials that we could take advantage of study and replicate thanks to bionics, but there are also special structures of certain biological surfaces. Some of the most famous examples are the lotus effect, lotus plant, Nelumbo nocifera, to produce ultra-hydrophobic self-cleaning surfaces, the gecko effect for the development of dry adhesion or surface, the mud age effect to achieve anti-reflective surface, and the shark skin effect to generate the anti-friction surface. Having said the possible applications of bionics in the study of these structures, it should be noted that not all examples can be studied by atomic force microscopy. This is due to great limitation, which is that this microscopy is restricted only to materials low roughness, so it is preferred to use traditional optical and electron microscopy. However, as we mentioned in the beginning, still developing such materials and the AFM is very useful to study the structure and properties of these new materials synthesized. Cryo-EM helps to study the parts that make up the cell from an atomic perspective, in such a way that bacteria and prokaryotic cells can be studied to obtain new traits that could not be found before. Bionics can be used to analyze the behavior and characteristics of cells and especially their components and the way in which they take advantage of them to subsist and interact with the environment. This which the purpose of replicating the functionality of cells to develop new solutions or technologies such as the structuring of material and organization of the molecules and atoms.
Hi, my name is Valeria and I will talk about how to form an AFM image. We need a simple with roughness on its surface. To measure this roughness, we use a tip that is placed close to the surface. We use a piezoelectric system called cantilever. When the sharp tip of the end of the cantilever contacts the surface, we find a deformation due to the roughness of the sample. This deformation can be measured by an optical system that has a laser and a 4-quadrant optical detector to measure this deformation of the beam in different modes. The laser boom is mounted above a piezoelectric table C to check the height and an X-ray table for the contact of the tip with the symbol. All the system has a computational measurement system. When the laser moves on the surface of the optical detector, a signal is generated and transmitted to the computer, resulting in a change in the table for height to keep the laser in the detector center position. The voltage required to change the height on the table is the signal coming out of the computational measure system. To measure the roughness, the surface is swept with the X-ray table and it can be done in different modes. These modes are four. First, contact mode. The tip is in contact with the surface and is placed on top of it for measure the contour. Second, intermittent contact mode. The tip vibrates in the resonant frequency close to the surface, coming closer to it while it increases the contacts. Third, no contact mode. There is no contact with the surface. The tip vibrates at some distance from the sample. 4. Force modulation mode. An intermittent force is applied. In this way, the tip penetrates the surface. And that's all for our team. Thank you.